Hello everyone and welcome to today's video. Today is part 2 of the cell, and today we're going to be going into all the cytoplasmic organelles. So everything from the mitochondria to the lysosomes to the peroxisomes. The next part we're going to be talking about the nucleus in the cell cycle. So right now these are just the organelles that are in the cytoplasm. Let's hop into it. All right, so first one here is everyone's favorite, the mitochondria. Now, the first ones I'm going to be talking about are the membranous organelles, or the ones that have a membrane. Some of the organelles in the cell do not have a membrane, and we'll talk about those second. All right, but first the mitochondria here. Everyone knows the meme. I hate the meme, but the mitochondria, you guys know it as the powerhouse of the cell. Why do I hate that? Because what's the powerhouse of a bacteria cell? A bacteria does not have mitochondria. Mitochondria is actually the size of a bacteria. However, there is an enzyme present in a mitochondria called ATP synthase. That's what the hydrogen ions pump down through and make ATP. Bacteria do have ATP synthase. So now we can upgrade our meme and now say ATP synthase is the powerhouse of the cell. The mitochondria does other things as well, but I don't like the definition of the mitochondria being the powerhouse of the cell because it isn't true throughout all biology. It's not the powerhouse of every cell, it's the powerhouse of eukaryotic cells. So a long, long time ago, mitochondria mitochondria, they replicate via binary fission just like bacteria. They actually have their own plasmid DNA in there. So here's circular DNA. They have their own ribosomes, which we're going to talk about later, which are the protein factories. So mitochondria are pretty much a little mini bacteria living inside of us, or a little unicellular organism that has made an amazing endosymbiotic relationship. So they make us energy, and we provide them a nice environment to survive. So mitochondria, that's where our ATP are made. So most of our ATP are made in the mitochondria. We make a little bit of ATP in our cytosol via glycolysis, but the full cellular respiration, electron transport chain, that is present in the mitochondria. For the most part, everyone knows the mitochondria quite well. Remember, it has all those folds to maximize that surface area. The next membranous organelle is the endoplasmic reticulum, or the ER. There are two different types of the ER. There is the rough ER, which is embedded with ribosomes. So there is a ribosome. That's where it gets its rough name. So ribosomes are the protein factories. So the rough ER is making proteins that are going to be secreted. So right here, this is this little vesicle being popped off and working its way down the secretory pathway, which we'll talk about later. And then right here, this is the nucleus. So the rough endoplasmic reticulum is right alongside the nuclear envelope here. They're actually continuous with each other. And then the other type of ER is the smooth ER, because it is not embedded with ribosomes. Now, what does a smooth ER do? Smooth ER is a major place for calcium storage, lipid synthesis, and well, so making and breaking lipids, so lipid metabolism, I mean. Also, it's an important area for detoxifying certain poisons and toxins. So the smooth ER is very important, but it doesn't get a lot of attention in Bio 1 because you all talk about the rough ER. But now the smooth ER, depending on the cell type and anatomy and physiology, becomes very important, especially if there's a cell that has a lot of calcium storage. Smooth ER plays an important role there. So we'll be mentioning that a few times throughout anatomy and physiology. So remember, our focus in talking about these organelles is structure and function. When we look at a cell in the microscope and you see a lot of rough endoplasmic reticulum, structure and function. Structure, a lot of rough ER. That tells you that cell is making lots of proteins. And we see that in something like the B cells in our immune systems, which are just pumping out antibodies. They're pumping out so many antibodies that actually commit suicide from running out of energy. But in those, we see tons and tons of rough ER. All right, next one now is the Golgi apparatus. This is, as you call it, the shipping and receiving center of the cell. So it's the UPS system. So vesicles pinch off the rough ER and go to the cis side of the Golgi, or the cis Golgi. Proteins then get folded and processed, and then go off the trans side. They go then to the trans side, trans Golgi. And now there are 3 different ways we can go after leaving the trans Golgi. One is exocytosis. Oops. So leaving the cell as a secretory vesicle. Another one is also kind of like exocytosis, but the Golgi, the vesicle contains transmembrane proteins. So just fuses with the plasma membrane, so just membrane proteins. The proteins, like receptor proteins that need to be sticking out of the membrane, are processed into Golgi and then sent there. So membrane proteins. And then the other thing that trans Golgi releases are lysosomes. These stay in the cell, and we'll talk about lysosomes later. They're the little garbage cans of the cell. So 3 different pathways kind of that these trans Golgi can send these vesicles. But in the end, they're packaged with proteins. They have transmembrane proteins, and these vesicles, we're going to talk about the cytoskeleton. One of the cytoskeletal components is called a microtubule. So there's these little motor proteins, and they kind of like sit like this and then attach to the vesicle. Remember, it's a phospholipid bilayer, and inside here we have all these proteins. So this actually kind of walks down the microtubules as we move that way using ATP. That's called vesicle transport. So when we look at these images, it looks like these little balls of vesicles are just magically moving. They're actually walking down the cytoskeleton, which we'll get to here soon. And then the next one here is a peroxisome. I decided just to write this one out here, then find an image. So whenever you see peroxisome, think peroxide. So what happens in our mitochondria is these are typically found right beside our mitochondria, and a byproduct of cellular respiration is sometimes free radical production. So what the peroxisome does is it takes free radicals and converts them to hydrogen peroxide, or H2O2, which in itself isn't safe, but it's safer than free radicals. And then it takes it one step further and converts it to relatively harmless water. So peroxisomes are pretty much our antioxidants, trying to reduce the amount of free radicals that could damage our cells, and then reducing that hydrogen peroxide it itself produces as a byproduct. So peroxisomes are quite important too. Lysosomes, these are then the waste management system of the cell, or they're like little stomachs. So this is a little vesicle, again, phospholipid membrane, comes off the Golgi apparatus, and it's filled with hydrolytic enzymes. So what happens here is that after it pinches off, it actually brings in hydrogen ions. So we get a high hydrogen ion concentration inside of the lysosome. That makes it acidic. So all these enzymes work in those acidic conditions. They're called hydrolytic enzymes, meaning enzymes that function in the presence of hydrogen ions. Now, what they do, so it's like a little stomach. This fuses with broken down mitochondria, fuses with food vacuoles, and helps digest certain components. An example of a disorder, a lysosomal disorder, is called Tay-Sachs disease, where one of these enzymes isn't made properly and they can't properly break down certain components. In the brain, I think it's even a bigger issue. So that's a lysosome, little mini stomachs inside our cell that help break it down. So those are all the membranous components. Now let's look at the non-membranous organelles. First one here is the ribosome, made up of 2 subunits. This is a small subunit, and this is the large subunit. Ignore all the rest in this. We'll talk about this in a later part when discussing the cell. So this is translation, and we'll get there, don't worry. But here, ribosome is made up of rRNA, or ribosomal RNA, and some proteins, and its purpose is a protein factory. So this is where all of our proteins are made. So right here, we see the polypeptide coming out of this ribosome, coming out of this ribosome here, and we don't have a protein in our body that's not made via a ribosome process and gene expression. Okay, so now we can move into the cytoskeleton. There are 3 components of the cytoskeleton, so 3 different size fibers. The first one here are the microfilaments, or sometimes I refer to these as actin filaments, because these are always made of this protein called actin that wraps around itself in this little double strand structure here. So these are little actin subunits that wrap around itself, and they are the smallest because they only have, they're 2 proteins wide. And these are important in, moving cells, so pushing the cell membrane and moving it around. Also, in our stomachs, the microvilli in our stomach, we have all these little projections in our not our stomach, our small intestine. And those extensions, those microvilli push out and they have actin in the middle. That way we can maximize our absorption area and bring in more nutrients for digestion. The next type here, we talked about these earlier. These are the microtubules. So microtubules are made up of a protein called tubulin. That's where it gets its name. These are the largest subunits, and they are hollow in the middle, and you can combine multiple of these together. And so a good example where these are used are in walking vesicles down. I just drew that earlier. So vesicle transport for secretory vesicles. They also help hold different organelles in place so they're not moving around everywhere. And also for cell division. Microtubules are what attach to the chromosomes and then separate them during anaphase and put them in the middle for metaphase. So microtubules, very, very important in our cells and part of the cytoskeleton. And the last part here, I also didn't find a picture for, but they are the intermediate filaments. Intermediate filaments are more of a structural support role. They're made of various types of proteins, but an example of one type of protein is keratin. And they're called intermediate filaments because they're in between the size of microtubules and the actin filaments, the microfilaments. So that's all I'm going to say about the intermediate filaments. They're just in between, and some are made of keratin. So more structure and holding things in place as well. And then here, the centrioles. These are the little microtubule factories. So centrioles come in pairs, and when they're paired up, they're called a centrosome. So during cell division, the centrosomes replicate, and they move to opposite poles. They start sending out their microtubules, and they connect to the chromosomes and pull apart. So centrioles are these little microtubule-making factories, and they're pretty cool, so I included them in here as well. And then with those, we could talk about the cilia and flagella. These are little, extensions, cytoplasmic extensions, where it actually can make things motile. So the one example of a flagella in humans is the sperm cell. That's how it moves. So it's a long microtubule or long strand of microtubules, and it has these power strokes and makes this propeller-like motion on the end of the flagella. So flagella are longer than cilia, and cilia are smaller or shorter. So cilia do little power strokes back and forth. Cilia are really important in us, especially in our respiratory tract, for moving mucus. So usually we find lots of cilia in mucus areas. And also in our hearing and balance, they're very important. And so there's a mechanical process that fires a signal for hearing and balance. And so when we talk about hearing and balance later in the nervous system, we'll come back to these cilia. They're actually embedded in these little stones, and it's really neat how that works. But here, they can move back and forth and move the cilia. And again, they're made of microtubules. Those are just much shorter and usually higher abundance than the flagella. Like a sperm cell only has one flagella, but they're made of microtubules, so really, really neat. So in summary, just going over the cytoplasmic organelles in this video. We're going to talk about the nucleus next part, so don't look at that right now. But we went over the plasma membrane last part. We went over the Golgi, ribosomes, rough ER, smooth ER, the actin filaments. There's a flagella, there's a centrosome, a secretory vesicle leaving the Golgi, and then the cytoplasm. One thing I didn't mention about the cytoplasm. A lot of people think the cytoplasm is this watery fluid. It's actually almost like a gel. So it's called a colloid, and it's a gel-like fluid, and a lot of people forget that. So it's very viscous. And then here are intermediate filaments. There's the mitochondria, free ribosomes, along with the ones that are embedded on the rough ER, then the lysosome, microtubules, and peroxisomes. So I hope that was a good summary of all the cytoplasmic organelles. And if you have questions, let me know, and I'll see you all next time. Thank you. Hello again. Thank you for watching my video. I'm glad you could come by. If you enjoyed this content, feel free to do that thing where you like and subscribe. Also, if you're interested, I prepare and record all of these videos live on Twitch. After the recordings, we do a post-recording question and answer section, and then sometimes I play games where any question is fair game as I game. Also, on Twitch, I'm a co-founder of a wonderful community called the Knowledge Fellowship, or TKF. Our goal is to seek and share knowledge with others while live streaming. We feature streamers who cover biology, law, physics, chemistry, geology, photography, health and fitness, psychology and mental health, rocket science and aeronautics, programming, and tons of different practical EDU streams. Feel free to check out TKF at the link in the description and join the very active Discord community. Hopefully, you find something that interests you. With that, I'll see you all next time and have a wonderful day. Remember to seek knowledge and challenge everything.